Hello everyone, this is Brainless Medico and I am Dr. Manoj, and today we are going to learn one more important topic under hematology, that is hairy cell leukemia. Important 5 marker, not expected for 10 marker, but important for 3 marker also, and MCQs for NEET point of view, it is very important topic since it has 2 important images you need to understand for your histopath part. So let's crack the next shell. So pathogenesis, why hairy cell leukemia actually occurs? It's a neoplastic proliferation of mature B cells. So there will be mutation commonly seen in BRAF V600E. BRAF V600E. You need to remember this. It is very important gene. Then coming to mechanism, there will be increased polymerization of G actin, which will be replaced by F actin. So there is a changes in the actin filament. So G will be replaced by F actin, which leads to cytoplasmic projections. So you can see here there are projections around the cell. So this is what happens. So because of this cytoplasmic projections, these projections give classic hairy appearance. So we call this as hairy cell leukemia. Then why splenomegaly? So there are some projections what it will cause, why splenomegaly will occur. So let's see why it occurs. These hairy cell infiltrates spleen. Splenic macrophages attempt to destroy tumor cells, so which results in splenic enlargement, hypersplenism or cytopenias. So specifically which cytopenias we'll learn in the further details. So massive splenomegaly is the hallmark feature. So when there is a massive splenomegaly clinical picture in the leukemia part, you have to keep this hairy cell leukemia in the option DDs. So coming to monocytopenia, as I said, there will be cytopenia. So first we'll learn why monocytopenia. Tumor infiltrates which immune destruction will leads to decreased monocytes. So we call that as decreased monocytes. We call that as monocytopenia. It is also a characteristic finding. So we know hepatosplenomegaly. Then next we have monocytopenia. Then coming to bone marrow involvement, since there is something related to blood related, so obviously the bone marrow has to be involved. So hairy cell infiltrates bone marrow. They stimulate stromal cells to release TGF beta. Remember TGF beta, not alpha, TGF beta. TGF beta increases reticulin formation. There is a image, histopath image related to reticulin formation. There will be threads like which will be seen under green color, but I felt like that is not that necessary to know for the exam point of view. Then results. So there will be marrow fibrosis, decreased hematopoiesis, and pancytopenia. So clinical features, as I said, massive splenomegaly, monocytopenia, pancytopenia, fatigue, recurrent infections. Then coming to how you are going to diagnose the condition, you have the clinical picture, everything, but you need a solid proof that this is the given condition. So you need to go with the further confirmatory investigations. Here we have bone marrow, aspirate we do, but we get there dry tap. Dry tap means there is nothing to get there. It is very dry. It's just a joke. Dry tap. Just remember it's a dry tap. Biopsy, we see fried egg cell, appearance of cells. So just see here. Consider this cell. It looks like a yellowish part and the whitish part, right? So it is called as fried egg appearance. Individual cell looks like fried egg appearance. Then coming to immunohistochemistry, we have best marker annexin A1. If this is positive, it's confirmed hairy cell leukemia. There is nothing else you need to remember. But for the exam point of view, they play with the other CD markers with you people. So you need to know if there are any controversies or something, you need to confirm with other CD markers. So we have CD20, 11C, 103, 25 and TRAP positive. Then coming to you got the clinical picture, you confirm the diagnosis, but final thing you have to treat it. So coming to the treatment, targeted therapy, we have targeted therapy. If there are refractory or relapse condition, we need to go with the other thing. So for targeted therapy, we have BRAF inhibitor since it is a mutation of BRAF V600E, right? So we have to target that actual condition so that it may be controlled, permanently cured, but it can be controlled. Vemurafin or dabrafenib. Then for refractory cases or relapses, we go with the rituximab with pentostatin or cladribine combination. These 3 drugs are given for relapse or what is a refractory. So in this scenario, we learned about hairy cell leukemia. It is a mutation of BRAF V600E. There will be hairy projection, so we call it as hairy cell leukemia. Splenomegaly, since splenic macrophages try to destroy it. So there will be splenomegaly, monocytopenia, Then we have clinical picture splenomegaly, monocytopenia, pancytopenia, fatigue, recurrent infections. Then we get bone marrow aspirate will be dried up, and biopsy we get side-eye appearance. Then anaxin is the main IHC marker you need to remember. If you want, you can read more about these. What do you say? Other specific, other positive markers. Then coming to treatment, you need to target the actual mutation gene, which is BRAF V600. So that's all for the video. I hope you people enjoyed the video. Don't forget to subscribe. And if you have any doubt, comment section open for the doubts. Thank you.